Hello and welcome to our first set of video notes. This is a pretty straightforward set. We're going to talk in here about different equipment and supplies that you're going to be using throughout the year. It's not overly complicated. Some of them I'm really not going to spend much time talking about because you know how to use them. A few of them I'll explain or give you some safety tips on using them. So a ruler is pretty straightforward, or a meter stick. The big thing about using meter sticks or rulers is to pay attention to the units that you're using and which side of the ruler or which side of the meter stick. Remember, in science, we use metrics, not inches. And so you're going to need to remember your conversions. Things like 1 meter is 100 centimeter. A millimeter is 1,000th of a meter. That kilo is 1,000 times something. So you're going to want to remember your conversion factors and to make sure you're using the metric side, not the inches. So microscope, we're not going to spend a lot of time today. We're going to do an entire activity going over how to use the microscope. But a microscope's used to make something that you have to look bigger so that it's easier to see stuff. There's different types of microscopes that biologists use. We use here in class light microscopes. They're the least expensive option and the easiest to use typically. But there's also some really cool microscopes that are electron microscopes. They take awesome detailed images. And then there's what's called a stereo microscope, which we also have here in class, and we may use them at some point in time. But we'll spend more time going over microscopes in detail and learning all these different parts and pieces and their names. So slides and cover slips, we use the slide to hold something, to put it underneath the microscope. The cover slip goes on top of it so that your lens doesn't actually touch your specimen. Be careful with the glass cover slips because they do tend to break easily, and you don't want to get any little shards of glass underneath your fingers. Trust me, it hurts. Goggles are necessary eye protection. If you're told to wear them, you need to actually wear them. They go over your eyes, not on your head or around your neck. As the Minions say here, wear your safety glasses to be cool like the Minions. If everybody wears them, then no one looks silly. Gloves are used to protect your hands. Gloves like you see here, these are for heat resistance. So their purpose is to protect your hands if you're picking up like a hot beaker or something like that. These are either latex or nitrile gloves that they're used to protect you against harmful substances. Here in class, we have nitrile gloves, and we use nitrile because there's not allergy issues that you tend to see with latex. Digital scale. Digital scales are going to measure mass in grams, so we can measure pretty small amounts with these. You can also use them to measure in ounces, but ours we use for grams. You want to make sure that you always click on this little 0 button, and that is to set the scale back to 0 before you put your substance on it to measure it at, so that there's no issues with excess weight, like a powder or something that's on the scale. Also, if you have a weigh tray, you want to put the tray on there before you put your substance on the scale to mass it, because the tray has mass to it as well. So graduated cylinders. These are containers that we use to do volume of an object. We read them in milliliters. Make sure you read the bottom of the meniscus. The meniscus here is the curve, and the bottom of that is the volume that you actually see. So take a second and figure out what's the volume that's represented on this particular graduated cylinder. Record that value in Edpuzzle for your question. So thermometers are used to measure temperature, and we measure in degrees Celsius. For, you want to, again, here you're going to be looking at where does the red line go to. So in Edpuzzle, I want you to record what is the temperature of the first, the second, and the third thermometer. Stopwatches are used to measure time. Most of the time what you're actually going to use is the stopwatch or timer on your iPads or on your iPhone. And the nice thing about these is they can measure down to the 100th of a second. So this is really handy to have your devices. This is a fun word. These are called scopulas. They're a combination of a spatula and a scoop, hence the term scopula. You got a pointed end and then you got a flattened end, and it makes a little scooper, and we use these to scoop up powders. Test tubes, they're used to hold small amounts of materials, usually liquids for different experiments. They're made out of glass, so you do want to be careful with them. If the test tube that you grab is cracked in any way or the surface of it is not nice and smooth, then let me know. We'll probably end up throwing it out. So the thing about test tubes is when you put a test tube in a hot water bath, the test tube gets really hot, and so you don't want to pick it up with your hands. That's when we use test tube clamps so that we can hold them without touching the test tube. You don't have to use a test tube clamp if you're just carrying a test tube around the room unless the test tube is hot. Really, we use them when things are going to be causing a problem for holding them with our hands. Okay, so test tube brush. Most important word on this slide. Test tube brushes are used by students to clean out dirty test tubes. You should not be leaving your dirty test tubes in the sink or in the bucket unless I tell you to. Test tube brushes get used by students to clean out the test tubes that they've used. They are down back by the sink. Test tube racks are racks used to hold a test tube. Pretty simple. These are great if you have multiple test tubes for something and you need to carry the multiple test tubes around the room. Rather than holding 5 test tubes in your hand, you put them in a rack, and it's easier to carry the rack around. Ring stands. We use these to support things with clamps and rings on them, like test tubes, beakers, or flasks. We probably won't be using ring stands too much, although there is one that is usually set up in my room, and it's associated with our distiller for water. And so you'll see one down by the sink all the time. Funnels are used to separate solids out of a solution, and you use them also to pour liquids into a container. So a lot of times we'll end up using the funnel to separate something, and so we'll put some filter paper or cheesecloth inside the funnel to separate the liquid from the solids. And hence you've got filter paper and cheesecloth next. So the filter paper is used to filter the substances. This is your filter paper, this is your cheesecloth, okay? And so we use both depending on what we're working with. Beakers are the fancy glass containers that we use to hold and pour things. It does tend to have a spout on it that's used for pouring and markings that can be used for measuring things. You do want to be careful if you've been heating your beaker that it can get hot. So you're going to want to use gloves for that if you have a heated beaker. And the other thing to remember is if the beaker's cracked or there's any chips in it, let me know. Likely we're going to end up throwing it out. Erlenmeyer flasks. These are triangular in shape. So their purpose is to heat liquids or temporarily store things. Like a beaker, they tend to have markings on them that we can use for measuring volumes on the side, but we tend not to use the flasks to measure out volumes. Typically using a graduated cylinder is going to be more accurate. Stirring rods are pieces of glass that are used to mix substances in a beaker. You do want to be careful. They're pretty sturdy, but they can break. If there's any chips on them, again, let me know. And if they're chipped, depending on where and how, then I just have to put them in a flame to melt them in and make them smooth again. Petri dishes are made out of, most of them that we have are made out of plastic, and therefore they're disposable. They can be used to grow bacteria, observe seeds that are sprouting, observe small animals. You can use them to hold little things, which is nice and handy. I love this picture here. This is a Petri dish that somebody put their handprint on. So you can actually see, here's their finger, there's their thumb, and each of their fingers. This is obviously a child's hand. And then here's the hand itself. So all of this stuff is stuff that was growing, that was on their hands. Spot plates are used to test bunches of little samples. We'll use spot plates throughout the year. The nice thing about these is that it makes it so that you don't use quite as much stuff. They can be a pain to clean though, because when you put water on them, they splash. So when you go to clean them, just be careful. Pipettes, these are plastic made. We use them to measure out small amounts of liquid like a milliliter, squeeze droplets of liquids, or transfer liquids. So the advantage of these is that you can get really small amounts. The disadvantage is that the droplet sizes are not exact. So if you're having to get perfect measurements, you're not going to be using a transfer pipette. Micropipetters, on the other hand, are extremely sensitive, and they are very, very accurate. The difference between these and regular transfer pipettes is micropipettes are measuring out in microliters. They're measuring tiny, tiny amounts. These are expensive devices. We will probably use them at some point in the year, but not as often as we would a transfer pipette. So this is a pipette that's going to transfer a single substance. These are used when you want to transfer a whole bunch of stuff at once. Hot plates are used to heat glass containers. You turn the knob to the different temperatures, and that's going to adjust your heat. You do need to be careful using the hot plates. You got to make sure you wear your goggles and don't ever touch the hot plate. Even if it looks like it's off, it might still be hot. Always make sure you check wires on them to make sure that the wires aren't frayed. I will check the wires before I put the hot plates out, but in general, it's a good idea to check to make sure they're not frayed. Okay, so this is a mortar and pestle. This thing down here is your mortar, that's your pestle. The pestle is used to grind things. The mortar is what you use, where you put the stuff that you're gonna grind or crush up. So usually we have something that's like solid, like maybe an antacid tablet, and we wanna crush it into powder. We're gonna use a mortar and pestle to do that. Dissecting pans. These are fun little trays that we use to cut stuff open. You want to make sure if you're using them that you clean them really, really well when you're done, because otherwise they get gross. The inside, this blue thing, is squishy material, and that's squishy so that you can pin things into the squishy material to hold stuff open. Okay, scalpels. These are used to cut stuff with, dissecting samples, not like cut a piece of paper. The thing is with scalpels is that you need to use caution because they should be really sharp. Dull scalpels and dull blades, like dull knives, are actually more dangerous to use than a sharp one. The dull ones, you're more likely to cut yourself. And so if we're doing any dissections, I always replace the scalpel blades at the beginning to make sure that you have a sharp blade. Dissecting pins, they look like pins that you use in your clothing. They're not. You use them to hold dissecting stuff open. So they're used to hold stuff in place or to hold open dissection stuff in when you're doing dissections. Sometimes they'll have like a little T-shape on them, and then you can use them to flag stuff. Forceps are used to pick up things that are really tiny or to pull pieces away during a dissection. So we'll use forceps a couple of times throughout the years for different purposes. Okay, dissecting probes are used to push stuff around, to dig under things or to point something else. Notice here that you have dissecting probes that are sharply pointed. This one has an angle on it, this one's straight. These have very sharp pinpoints on them. You want to be careful with them. If you're digging things, or whenever you possibly can, you want to use the one that's dull, and that's so that you don't actually damage the sample that you're looking at or stick yourself with it. Okay, dissecting scissors are different from regular scissors. Dissecting scissors are used during dissections only. They tend to be extremely sharp, and so we're using them to cut stuff away. You don't want to cut too deep because you could cut something you don't mean to, and you can't uncut something afterwards. So multiple shallow cuts are always better than a few deeper cuts when you're doing dissections for things. You want to be careful with them. Like I said, they're sharp, but we do only use them for dissections, so it's not like they're going to be out whenever we're doing stuff that you have to cut. If you have any questions, make sure you write them down and ask when we see each other in class next time.